You're welcome once again. You're welcome once again to another class. Madam Shadi, please, you may have to put your mic on mute so I won't hear the background noise. Hope you can all hear me. Can you all hear me? Am I audible?
Yes.
Hello? Okay, good. Okay. So you're welcome back from the long break and hope it was refreshing. Hope it was refreshing. So we have covered quite a lot in this course, and I will say that I'm impressed with the performance of your response. Even though it would be better because I have over 100 people that are doing this course. That's the information I have, over 100 people. But the fact that they are still showing up is still something. So today is going to be the 6th week, and I don't know. Most likely, we should be able to have 1 or 2 weeks more for the exam. 1 or 2 weeks more. Sorry about that. That's a network problem. That's a network issue. Okay, so like I was saying before, if time is so close, then that means you have to choose a day where we can just quickly do our revision and know what we have done so far. For some of you joined later, I didn't start at the beginning of the course. So we'll choose a day for that. But I think we should still have 1 more week or 2 for your exams. So everything is still dependent upon you people, how convenient it is for you. Okay, so moving on. My name has not still changed. I'm still your facilitator. ALAWODE Yetunde is my name, and the course is CRP305, crop genetics and breeding. So, like I said, today is the 6th week, and we'll be talking about steps for sourcing. Okay. Steps for controlled crosses and sourcing of breeding materials. So we want to know the steps of crossing and breeding materials. Most of these things are usually practical in nature, but I just want to let you have an idea, because I will tell you to state the steps for me, explain the steps for controlled crosses and sourcing of breeding materials. So plant breeders, of course, depend on existence of genetic variability to select superior genotypes. That's what selection and plant breeding is all about. superior genotypes, genotypes that are better than the ones that you can get in the field or in the wild. So what the plant breeders do is just to keep developing more varieties that are better than the existing ones, and they can only get it from the variability that is available. You can only work with the materials that are available. So that is what they do. They take the materials from the wild, from what is available, and then they develop varieties from there. So the main way or method of developing these varieties or creating new variabilities by making crosses. Is by making crosses. That's the method. So what are the steps for controlled crosses? There are 5 steps here. And the first one is the choice of the parents. Of course, that is very important because the choice of the parents is the one that will make you know what the offspring will be like. Like I've been talking about all the while, the parents are important. And when you talk of parents, 2 parents are usually involved in the crossing. You have the P1 and the P2, the male and the female parent. So after you choose your parents, the next step is emasculation. And the next one is pollination. Next is tagging. And then finally harvesting and storage of F1 seed. Of course, you know that what you get from crossing your parents is the F1. Once you do when you cross parents together, the seeds that you get is the F1. That's the first filial generation. Then when you go ahead to self, then you get your F2. So these are the steps for controlled crosses. So we'll be taking those steps one after the other. The first step we'll be talking about is the choice of parents. And it's very important to know that when you are choosing your parents for hybridization, you have to make sure that you are certain of the homozygosity of the parents. The parents usually should be homozygous in nature. It is when you get your heterozygosity that you now know that you have to self. But most of the time, the choice of parents are usually homozygous in nature. By now, you should know what homozygosity means. And you should also know the agronomic base of your parents, of the parents that you are choosing. What is the agronomic? What is their agronomy about? What kind of environment are they well adapted to? What kind of climatic conditions are they well adapted? What is the agronomic base, the soil type, everything that's got to do with the agronomy? These are things that you have to know when you are choosing the parents of your crops. So, like I said, the parents chosen for hybridization must be in homozygous condition. And usually when they are homozygous like that, they are pure. They call them pure line because they are not contaminated. They don't have any other traits that is contaminated, that is hidden. They don't have traits that is hidden. So they are usually homozygous. They are pure for that trait. And yeah, I told you that the agronomic base is talking about the area where the parents thrive the best, where they are well adapted to. So those are the 2 things that you should know about the choice of parents. And the next point is the emasculation. Emasculation simply means removal of male sex organs of the flower without damaging or disturbing the female reproductive organ. So that's the next thing you do. You have to emasculate. When you plant your parents, the next thing you do is to emasculate. And when you are emasculating, you are removing the male sex organ. You don't want pollination to take place. especially in self-pollinated plants. You don't want them to pollinate themselves. So you have to remove the male sex organ without disturbing the female, because its female doesn't have to be tampered with, because you are going to go and bring another male from elsewhere, from another plant, to come and fertilize that female. So the emasculation should be done carefully, such that don't disturb the female reproductive organ. When you are doing it, you can see in this picture here, the man's hand is steady. Somebody's hand that is shaking cannot do that kind of thing, because you make mistakes. So these are the things that you learn as a plant breeder. And it is very much essential to emasculate the plants to avoid self-pollination. Like I said, you don't want that, because if you let the plant pollinate itself, you are getting another one of that plant. You are getting another pure line. But what you want to do is that you want them to cross. So they are pure line, and then you emasculate so that you can go and bring pollen from another plant, and then you put it on the stigma, like I was explaining in the last class. So there are different types of emasculation. There are different types of emasculation, and the first one is hand emasculation. So the general procedure for hand emasculation is that the first one is emasculation is done before the anthers mature and before the stigma becomes receptive, because this pollination process is so fast. It's so fast that you may not even know that pollination has taken place. Then you are now busy emasculating, not knowing that the pollination has already taken place. So you have to do it before these organs mature, because the moment they mature and pollination occurs, you will not know. It's when you now see that the thing grows, you now see the F1, that you now know that this is not what I'm expecting. You see that pollination has taken place. But when they are not mature yet, then there's no how pollination can take place. So to minimize self-pollination, you have to make sure that emasculation is done before the anther mature and before the stigma becomes receptive. And also, the B part of this hand emasculation is that the petals of selected flowers are open with the help of fine tip forceps, and the anthers are removed. That's how the emasculation is done. Like you can see in this picture here, you have to have your forceps. Your forceps have to be clean and sterilized. Most times you put your forceps in alcohol. Like when I say alcohol, I don't mean the alcohol you drink, or that you're going to be taking beer and you'll be putting forceps inside. That's not what I'm talking about. You are talking of spirits. This is your methylated spirits. That is it, methylated spirits, not for those of you that drink spirits. That's not what I'm talking about. Methylated spirits is the best form of sterilization liquid that can be used. So what you want to do is your forceps are clean. You dip them in spirit so that it is sterile. So you now carefully open the petal of the flower. Carefully open it, because you have to do it before these things get mature, like I said. So most times when the flower is not mature, it doesn't open. I spoke extensively about flowers in the last class, so I'm not going to talk about it now. So you open the petals, see how the flower bud was before, see what it is now when it is forcefully open, and you can see the forceps. So what they are doing is that they are removing the anther. This is what the anther of this plant looks like. They will remove the anther, and then they will leave the stigma alone. So when the stigma is mature, they will now bring another anther, another pollen from another plant, and use it on it. That is how hand emasculation is done. For the suction kind of emasculation, you use this suction method when the flowers are very small. For example, the flowers of tomato. Tomato flowers are very small. That's why most of we plant breeders use glasses, because you have to look at things that are tiny. Sometimes you have to even hold a magnifying glass like this before you can see the flower, before you can see the anther of some of these flowers. So it's not an easy job. And it's something we do over and over and over again, because depending on the method or depending on your expertise, most times you just get the result of maybe 10%, 5%. You are not so good in it. So you have to be doing it over and over and over before you become an expert in it. So in the case of this suction method, it's used when the flowers are very small. The petals are generally removed with forceps and exposing the anther and the stigma. Then a thin rubber tube or glass tube is attached, okay, to a suction hose. So what that suction hose will do is that it will suck the pollen grain from the surface. So that's what the suction does. So when you open your flower, your petal, you now attach, and you have to do everything carefully. The rubber hose is thin, and what it does is that it will suck the pollen off the surface. For the hot water emasculation, just like the name hot water, you can use hot water to kill the pollen. So pollen grains are usually more sensitive to hot water than the female reproductive organs. So what you're doing is that you are exposing this treatment to, when I mean hot water, I don't mean boiling water. If you use boiling water, you kill both the female and the male flower. Something that is hot, just between 40 to 44 degrees centigrade of water for 10 minutes. You can use that, and this system is effective in rice. But of course, because of the hot water nature, it is not so effective as such. Then you have the alcohol treatment. Like I said, when I say alcohol, I don't mean your beer, your 3-3, your whatever, your Legend, whatever. No, that's not the alcohol I'm talking about. I'm talking about your ethanol. Okay, there are so many forms of alcohol that you use for all these lab processes. So this alcohol treatment is not a popular method because of the concentration of the alcohol, So the concentration of the alcohol for some time can, maybe when the pollens are exposed to alcohol at certain time, maybe for a few seconds or so, it can kill the pollen grains. So you have to expose them a bit. It's a very careful and tedious process. So it's not a popular method, because most times eventually they end up killing the female as well. So exposure to a bit for a few seconds is recommended, so that you will increase the female receptivity. You can also use cold water treatment like the hot water. Extreme cold water, extreme temperatures could kill the pollen, okay, without killing the female organ. Like, for example, in rice as well, when you expose them to between 0 to -6 degrees centigrade, then it kills the pollen. But of course, this system as well is not so effective. It's not so effective. So we have the genetic emasculation, like we talked about in the last class. You can use male sterile lines. You know that those ones, they don't have viable pollen. So you can use them for your breeding work. All you have to do is that even if they are there or they are not there, they are not functional. So you just go and bring another pollen and then use it for the female organ that is present in your genetic cytoplasmic or fluid sterility variety. So genetic emasculation, the kind of emasculation method as well. You can see that in sorghum. So after you have done the emasculation using any of these methods that I've already listed, I listed quite a few and I explained what they are all about. The next thing you have to do now is pollination, because this system, like I listed it, that is the way it should be. So it should be listed as such. It's not that you will go and put pollination first, then you will now put tagging last. It follows. It has to go in succession like that. So you have to, when you are writing it for me, you write it in succession and you explain it as such. So the next process or the next stage is the pollination stage. And you are not new with pollination now. You all know what pollination is about. Mature, fertile, and viable pollen from the donor plant. That is the plant that you are bringing the pollen from. You as a breeder, you know what you are looking for before you select your parents. It may be wide leaves, maybe for vegetable plants. You know that the edible portion of the vegetable is the leaf. So you may be looking for wide leaves. You may be looking for maybe stem, maybe it's the fruits like the okra, for example. People eat the leaves of okra, they eat the fruit as well. So depending on what you are looking for as a breeder, before you select your parents. So right now, you have emasculated the male and you are bringing in another male to come and pollinate the female. So your mature, fertile, and viable pollen from the donor, the place you are bringing it from, should be placed on the receptive stigma of that plant that is emasculated to bring about fertilization. Okay, so during pollination, pollen viability is a major factor, as well as the stigma receptability. And detailed procedure pollination varies in different kinds of crops. The way you do pollination in maize is different from the way you do pollination in okra. It's different from the way you do in tomato. It's different from the way you do in cowpea. So depending on the kind of crop, then you use the procedure based on type of crop, because they don't have the same flower. The flower in okra is bigger than the one in tomato. The flower in cowpea is a bit bigger than the one in okra. No, the one in tomato as well. The way maize is, the way the male, they don't, maize don't produce flowers, but you know the male organ, you know the female organ. So depending on the crop, we know the pollination procedure to use. After pollination, you must tag. You must tag immediately. You don't have to wait till for a second before you tag. Okay? And tagging is simply putting a label. You are tagging that cross that you did. So the emasculated flower is tagged. And this tag is usually made up of lightweight thin plate and is written with carbon pencil. This is what a tag looks like. You can see it has a rope like this. It is lightweight. So the plant doesn't even feel that any tag is on it. It's not that you put tag that will pull the plant down and will not be able to get, you won't be able to get your result. So it is just lightweight and it is thin plated, because when you write on it like this and the rain comes, if the rain comes and washes it, it doesn't affect this material because it's a special kind of material. And you write with pencil, you don't write with biro, because it's only pencil that will make it to be clearer like this. So once you do your cross, you have to label it, you tag it, so that you'll be able to know when it is fruiting, you know what this tag means. So that's tagging. And what information should be on your tag? What are the information that should be on your tag? The first one is the date of emasculation. Like you can see in the tag that I showed you, there's a date there. It says 2nd of October 2012. That's the date of the emasculation and the date of pollination. Okay. Yes, in some places, in some crops, you don't do the pollination and emasculation on the same day. So you have to, because if you remember very well, I said that you have to do the emasculation before the anther gets mature or the stigma is receptive. So you may do it a day or two before that. So you have to write the date of the pollination and emasculation, and the dates of the the names of the male and female parents that are involved in the cross. You can see a name here. There's this S something 33 by this times is a sign of the cross that is done. So you can see the two parents here. This is the first parent, this is the second parent. So that's what should be on your tag, as well as the name or names of the pollinator. You yourself, you have to put your name there. It's not something you put now, just put it, you just put your initial there. Okay, it has to be on your tag. So those are the things that should be on the tag. And the next thing you do after tagging is bagging. So after pollination, the flower is enclosed in a suitable bag so as to prevent random cross-pollination. That's why you do this bagging. So this is what the bag looks like. It looks like this your paper bag that you normally go when you go to places like ShopRite, you want to buy your chicken, you know, or your food. Those type of bags, not the nylon bag now, not the yellow bag. When you go to the food section, you want to buy your roasted chicken or something. That kind of bag that they give you, like a paper bag. Okay, so that's the kind of bag that they use. And the bag is usually thick, but not too thick. It's paper in nature, but inside has been lined with like nylon, so it doesn't get wet. Even if rain falls on it, it doesn't get soaked, it doesn't get wet. Okay, so it's like weatherproof. So that's the kind of bag that you put on the cross. You put it on it like that. If you see where they are doing it, you can see it in the picture here. Just put it, this is what the bag looks like. Put it on the cross here to prevent cross-pollination, random cross-pollination. And as the moisture and temperature become higher within the bag, you know you are closing the plant. So it will build up moisture, and also fungi may begin to develop. That is why you have to remove the bag after 2 to 3 days. 2 to 3 days after pollination, you remove the bag. Okay? It doesn't have to be there forever. By 2 to 3 days, you have assumed that pollination has taken place. So you have to remove the bag from the cross. Okay, so how do you source for, after bagging, you have to source, how do you source for breeding materials? How do you source for breeding materials? You can see on another farm here, you can see that the maize here, all of them have been bagged. Okay, they have been bagged. It will be an advantage to look for materials that are already adapted. And the steps to take in getting your materials is, first you look for commercial varieties that are available. You go to the market. When you're talking about commercial varieties, what is in the market? If it is maize you are looking for, go to the market first. If it is cowpea, go to the market. What do you have in the market? What are the things that are being sold in the market? Go and get some materials from there. And old varieties from farmers. Okay, you can also get your your varieties from the farmers. Then you can also get from breeders, breeding lines for breeders. And you can also get from National Center for Genetic Resources and Biotechnology, that's NAGRAB. You can get your materials from there. You can also select germplasm collection from your area. Just go around your area. Whatever you're looking for, you can get it in your area as well. And you may also get from other countries. You can source your materials from other countries where the same crop is cultivated. So these are places that you can get your varieties of your breeding materials from. So after talking about that now, I've talked about the steps that are involved in crosses. We've talked about all that. We want to take another new topic, which is selection. Selection and selection from the name. Well, when you talk about selection, what comes to your mind? Can somebody just tell me what comes to your mind when you talk about selection? What comes to your mind? Can you tell me so that I won't be the only one talking? Yes, what comes to your mind when you're talking about selection?
What comes to mind is picking a preferring something over other.
Yes, yes, you are choosing, right? You are choosing. So when you choose, you are making a choice, you are selecting one over the other. Okay, so you are choosing. That's what selection is basically about. So the ability to recognize superior plants, okay, in a limited array of variability. So you have a lot of crops, you have a lot of species. So the ability to be able to recognize and choose the superior ones around them, that is selection. And this selection came about by a scientist, a biologist called Johansen in 1926. And is the one that defined pure life as a progeny or the baby of a single self homozygous individual. So a pure life is the progeny. When you talk about progeny, you are talking about the baby, the offspring of a single self homozygous individual. So in the case of pure line, the genes are fixed. They are fixed. If you plant them for 10 years, you keep getting the same results because they are fixed. You can't see any change. That's why they are called pure line. They have reached the peak of, you can't see any change. Okay, if at all you see any change, any little change, change is due to environment. It's not due to the genetic makeup. So that is what selection is about. And there are methods of breeding self-pollinated plants. The method that you can use to breed self-pollinated plants, you can use your pure line selection, your mass selection, or your hybridization. So in this class, we'll be talking about pure line selection and mass selection. Maybe we'll talk about hybridization in the other class. And what is the method of using pure line selection to improve self-pollinated crops? You know what a pure line is, and so you can use it to improve on your self-pollinated crops. You know what self-pollinated crops are as well. So what is the method of using this pure line to improve your self-pollinated crops? Before you can get a new variety, it cannot take you anything less than 9 to 10 years. If at all it is going to be fast, it can't take you anything less than 7 years. Let me just put it that way. It's a lot of work and tedious work. But when you get it, then it's usually very, very good. So you are talking of like a minimum of 7 years, it could be more. 8 years, 9 years, 10 years, depending on, before you can say you have achieved, you have gotten a new variety. So for the method of using pure line to improve self-pollinated crop, the first year you have to make selections. You know what selections are now. So you have a heterogeneous population that is mixed with all manner of individuals. If it is okra you are looking at, for example, you have a lot of okra species. You have some that are long, some that are wide, some that are so different, and many like that. So the population of the okra is heterogeneous, so many. So as a breeder, the first thing you have to do is to make selection. You make selection. That's what you will do the first year. So in the second year, you will now do what is called progeny testing. So what you will do is that you will now select the seeds from each individual that you have chosen in the first year. You plant them out. You plant them in a row. So each row will stand for each of the selected crops that are grown. So you call it progeny row. And you now evaluate the plant. The ones that you like, you keep them. The ones that you don't want, you eliminate them. So as they are growing like that, you know the ones you want, you know the ones you don't want. So you keep selecting like that. The ones you want, you keep them. The ones you don't want, you remove them. So you now, out of the rows that you have planted now, you now select the best rows. Maybe you started with 10 rows, for instance. You select the best. Maybe you select like 6 rows, and then you now harvest all the rows and put the seeds together. So you now identify for experimental strains. You identify the lines you want to use. That's what you do in the second year. So there are 4 things you do in the second year. You take your seeds and then you plant them in the progeny row. And in the progeny row, you evaluate the plants and eliminate the bad ones. Then you select, you harvest, and you identify those lines for experimental strains. So most of these things is something you can read on your own. The third year, you keep repeating what you did in the second year. You plant again when you have already selected 6. Out of those, you know you started with 10. You eliminated 4, you left 6. You will now plant those 6 again. Plant them in a row again. You keep selecting the good ones, keeping the good ones, eliminating the bad ones. You keep keeping the good ones, eliminating the bad ones. That's what you will do in the third year. The fourth year, you keep continuing and observing and selecting again. Then when it gets to the 5th or 6th year, you do what is called replicated yield trial. Okay, replicated yield evaluation. So you plant them in replications. That's what they call, you repeat. You have maybe 3 or 4 replications, okay, in different locations. So you want to know their performance across different environments. So when you now do that, you finally select again. You keep selecting for the best, the one that performs well in the best in all the environments that you have chosen, the ones that are good, the ones that can withstand pressure. You keep selecting till you get your best variety. So once you have identified that, you now increase that variety and you release it as a new variety. So you can be sure, because of this test that you have done for, this is like the 5th or 6th year, like 7 years, you can now tell the farmer you want to sell your seed to, that you are sure that this seed will perform at so rate, because you are the one that did your selection, you are the one that know how the performance of the seed is. And of course, the variety should be uniform. When you plant it, you should get a uniform result. So that is for pure line in self-pollinated crops. Now for the mass selection, Mass selection is a breeding method that improves the general performance level of population by selecting and bulking. They call it mass because you are selecting so many individuals. Okay, unlike the other one, you have to eliminate a lot. But this one, in mass selection, you are bulking. You are getting as much as you want from the population, as much individuals as you want from the population. So in mass selection, a large number of plants of similar phenotype are selected and their seeds are mixed together to constitute a new variety. And what are the objectives of mass selection procedure? The first one is the improvement of local varieties. What mass selection does is that they improve local varieties. And these local varieties, of course, they differ in their flowering, in their maturity type, disease resistance, so they improve upon it. And they also purify existing pure line varieties. That's what mass selection does. Also, production of new varieties from heterogeneous local land races. These are the objectives of mass procedure, mass selection, and also these things are not difficult. The first year you select as well. That's the method of mass selection without progeny testing. I've explained what progeny testing means. Like you are planting your progeny in your line, that in a row. Whatever seed you get from the parents, you plant them out. That's the progeny. And in the first year, you select plants based on their performance, like whatever you're looking for. It may be vigor, it may be disease resistance, whatever you are looking for. You select plants based on that. When you plant your crops and you go to the field, you see that even with the insects, some plants will still do very well. You can select that kind of plant. And that means that that plant is tolerant to that disease or that insect. There are some that when you get there, the insect will not even come near the plant. You will see that the insect are eating other plants, but this particular plant, this insect hasn't touched it at all. You select that kind of insect, because that kind of plant, because that is what you want. As a breeder, you have to have a breeder's eye to be able to look at things like that. So when you now select, you now harvest them and you bulk them. That's what you will do in the second to the fourth year. Then you get your bulk seeds and then you get a new population. So what are the advantages of self-mass selection? Number one is simple to follow. New varieties can get to the farmers quickly, and then it reduces time and effort because you don't need any kind of extensive form of testing. And since a large number of plants are selected, the variety is usually more stable in performance. And traits that can be best used in mass selection are traits that you can easily observe, okay, by visual observation. Like I said, you have to have a breeder's eye. Whenever you go to the field, your eyes has to be at alert all the time to be able to see what you want. And also, you select for traits with high heritability value. So, having said that, I think we have come to the end of today's class. Any question?
Yes, ma'am. We have questions.
Okay. What's your question?
We are it's the general class actually. It's not just it's not my question. We are going through some past questions, so we came across some calculation about the heritability, this narrow sense, the broad sense. So there was this question. They asked us they give us data. They asked us to calculate the narrow sense and the broad sense. First, yes, we know that the formula to get the broad sense. It's
Okay.
it's a VG over VPH. So in the data, we know that to get VPH, you have to sum up the VA plus VD plus VI, right? So we don't have to calculate it because they give us VPH in the question. So my question here is, are we going to calculate that and forget about the one they gave us, or we are going to forget about the VD? Because the VA, VD, and VI, they are in the question, and the VPH as well. So which one are we to pick? If for example, we get that kind of question in the exam.
If the VPH is there, then there's nothing for you to calculate again. Since they have given you VPH, right?
Yeah.
So there's nothing for you to calculate again.
Okay, we should just ignore the VA, VD, and we should just ignore them.
Let me see the question. Send it to me on WhatsApp. Let me see the question. I don't know what you're saying, but let me just see the question. Or to me, or if they give you everything, then there's nothing for you to look for it. You just substitute your values for it. Any other question?
No. And sorry, yeah. This is the same. Is the same past question as well. This one, they say that we should tabulate the ask us to do the hybrid cross.
Okay.
They didn't give us any letter, so we know that we are to use we are to use any letter that person can use. But what is there is the access to tabulate. We don't understand. We don't actually understand.
That PUNET diagram is the, I think that's the tabulation.
Okay,
let me see the question as well.
That was my assumption, but
that was my assumption. What they mean.
Yes, that was my assumption. So
we just PUNET square.
Yeah, that was my assumption. So we just I just say let me bring it up so that we'll not make mistake.
I think that's what they mean by the PUNET. You tabulate. You know that PUNET square is like a table.
Yes.
Okay. Any other question?
No.
Let me ask my own questions then. Who can tell me the steps for
I sent the past question. Yes.
To me on WhatsApp.
Yes.
I'll check it later. So Mr. Job Adeli, can you unmute yourself and tell me the steps in controlled crosses? Controlled crosses, yeah, steps for controlled crosses. Are you there? Adeli Job. What about Okoronko Christiana? Anyway, I wish you people the best too, because okay, I'm listening. What are the steps?
No, I said I just joined.
You just joined.
I don't.
You're welcome. Okay, Shade, you have a question?
Yes, ma.
Yeah, what's the question?
The previous one to talk them about. I was not in the class, so later I listened to the audio. Then I was not understanding the way how to calculate the ratio.
That's why it's good to be in class. Now that one is you people that will find a way of communicating and explaining to each other. I'm not going to talk twice. I'm not going to talk twice. You just do your, that's why I said I encourage you people to do what is called tutorial. When you do tutorial, you explain to each other how, most of you have done the assignment and you have submitted and have marked it. So just get in touch with other members of the class and do a tutorial and do whatever you have to do to study. That's the best I can say, because I can't go back and repeat myself again. It's not convenient for me. Let's say we were in class now and then you told me you didn't understand this, I will have explained it. But in this case, you have to get in touch with your people. Speak to Farouk, to Gloriedet, and even Shadi. I think they submitted assignments to me. Even Joe Badini too. I saw the assignments and I marked. So tell them to explain to me. Okay, it's 2 minutes to 11 now, and I don't think Shadit is online to list the steps in controlled crossing. You want to say something? I can't hear you.
Yes, ma'am.
Okay.
The step involved.
Yes.
For control process. Yeah, first one, choice of parents. We have to choose the parents.
Okay.
Then
and when you're talking about choice
emasculation.
of parents.
Yes. Emasculation. That's the second one. Third one, pollination. Yes, pollination.
Yes, pollination.
Then we have to tag, labeling, tagging. So the next one is harvesting. You have to harvest and store the first one, first generation seed.
Okay, yes, F1 seed. Okay, yes, those are the steps. So you should be able to explain them as well in case you see them in the exam. So it should not come as a new thing to you. Okay, so it's 11 o'clock already. Please try and read. People have each other to be able to come. Yeah, like I said, we'll still have one more class. But before that, I don't know what your timetable is. Your timetable finally out now? Because I still need to do one revision class before you go to the exam. So let me know what your timetable is.
The timetable is out, and exam will start on 25th.
On the 25th, right? Yeah. When is CRP305?
27th.
27th. See?
Yes.
That we don't have any time now. So it's better we have 2 weeks, 2 classes next week. It's better we have 2 classes next week. I will do one more class, then we'll do a revision of everything we have done. So we have our normal class on Wednesday, and maybe Wednesday evening, we'll have another class for another revision. So you people should just prepare like that for next week. Next week, Wednesday is what date now? Day is 12. So next week will be like
19.
19, right?
Yes.
Okay, so 19 is still good. You people should just prepare for 2 classes on that Wednesday. We'll do 1 class in the morning, our normal 10 to 11, and probably 1 class in the evening when you people are at home, maybe like 6 to 7. Okay, just a revision class before you go for your exams. So,
thank you.
In the absence of any question, meet tomorrow for CRP303.
Thank you.
Thank you.
All right. Have a nice day.
Thank you.